All right, welcome everybody to the Hackathon Showcase. This is a moment we've all been working towards, and everybody's been very enthusiastic, very hard, even up to the very bitter end. And we appreciate all your efforts. Probably going to run over a little bit. I hope that's okay. If you have some kind of conflict, let me know. If then and somebody's late, then people can switch to a different time. Wonderful how much participation that we've had. Let me go to the next slide. Okay, and here's our agenda. So this is on also on Airtable. That's been updated pretty frequently. So the ground rules for the hackathon are that you have 10 minutes per presentation, or I should say you have 8 to 10 minutes for your presentation, and that includes question and answers. And the judges have expressed that question and answers are very important. They really want to know that it's not just pretty, but that you know what you're talking about, and that you know that the impact it's going to have and how much thought you put into it, and not only just the coding. So I really urge you to try to budget your time, leave a little bit of time for questions. I will be giving some warnings when you're getting to 8 minutes and 9 minutes, so you know that you need to wrap up and allow some time for questions. So here's the order of the presentations. I've made it random, so it was random order, just so you know, kind of be fair. You got the time, number, name of your team, the project title, and the team members. I apologize if anything is mistyped or is not up to date, because I know things were changing a lot. If you reach the 10-minute mark, then going to have to ask to move on. I know that that's tough, but we need to make sure there's time for everybody to present and everybody gets the same amount of time. You know, there was talk about making this 3 hours, but I think that would be really long. So with that, I think that I will open it to Team Alpha. So is Zhong Liang presenting, or everybody's presenting?
Everybody.
Okay.
Yep, we're all here.
Yep.
Yep.
Make sure everyone's microphone's on. Yeah.
Okay, we are good, I think.
Yep, good. Good morning, everyone. We are the Team 6 Alpha, and our group member are Clark Xu from University of Wisconsin Medicine, Dr. Zongliang Yue from UAB, Vanessa Xiao from Park Tudor High School, Vasileios Adavazos from University of Egin, and our mentor is Dr. Dick Chen from University of Alabama at Birmingham. And our title is Conjunctival and Corneal Tumor Detection and Recognition. So as you can see in the abstract figures on the right side, we intend to have we intend to collect the image data from patients, and we try to automate the diagnosis with a high accuracy, improve healthcare providing and disease monitoring. So why we care about this type of the melanoma? Because this is a rare disease but deadly, and it's occupied over 3,000 adults in US annually. And despite the excellent macroscopic excision of this mass, there's still 26% patients will develop in 5 years and 65% in 15 years. And we also searched the literature about the biological relevant. It seems that there's not a lot of the biological relevant, but a little relevant may be the age. The elder people will have a relative high risk. And in the genetic study, they report there's 3 mutations genes, but on the third promoter mutation was significantly shorter as compared to non-third promoter mutation in tumor. As you can see, this type of the melanoma have specific conjunctival region has this mutation and will yield a tumor. And with the timeline, you can see in the early stage, the conjunctival can have appeared in the nervous with a 10 or pigment, but afterwards it can develop acquired melanosis that cover afterwards it can grow up to a small or a large. So our hint is earlier diagnosis, the better treatment. And since the genetic study doesn't help a lot in the diagnosis, we're going to shift our focus on image data.
Thank you, Jean-Claude. So our application focuses on detection and recognition of early stage of the development of tumor, where machine learning models could help ophthalmologists to easily understand and control the growth of cancer. Next slide. To accomplish this goal, we have to work on supervised and unsupervised model in order to create a robust pipeline. Unfortunately, for this hackathon, we worked on a small dataset of 445 samples and 3 classes. And on the right side, we can see the parameters where we tried to replicate it from a paper, and we used pre-trained data from images. Next slide. Next slide. Thank you. Now, moving on to the basic notion of these models, we choose to pick the VGG19 because it was the deepest and most complicated model of this type of family, and that resulted in a decrease in accuracy. And because of small data, because of the small amount of data, there is an overfitting problem. EfficientNet, on the other hand, is a new approach to convolutional neural architecture with scaling method of depth, width, and resolution dimensions in the convolution network. And using too deep network will reduce the accuracy. Now, ResNet focuses on identifying shortcut connections in order to avoid the gradient problem. And finally, exception is based on inception, where the basic idea was stacking convolutional layers, not only vertically, but horizontally. Next slide.
According to our empirical results, we have observed that we have used the 4 deep learning models and compared to the baseline using the k-means. As we observe that the efficient net B7 is the best, as they have the highest accuracy in the training side and testing sides. In additional dimensions such as the structure simplicity that we have measured how many seconds per epoch, as we suggest that the efficient night B7 is the best accuracy, so that it should be the best to perform. If you have the limited computational sources, you can choose VGG19 or exception.
So during the course of our project, we encountered some challenges which we had to work out to get our models to run. So the first one was a small data set size and also some missing metadata, for example, the genetic data and the timelines on the images. So we resolved these issues by doing data preprocessing to transform the right features we needed for our model. And because some of the metadata was missing, we also tried out unsupervised learning techniques, such as different types of weighting to improve our training and test accuracy. And then another issue we had with the competition was time constraints. So typically hyperparameter tuning takes a long time in terms of processing CPU runtime. And in order to have the most efficient type of hyperparameter tuning, we use grid search, which is a very straightforward method of looking at different hyperparameters, such as iterating over epochs of 8, 15, or 30. And then we also used a machine learning algorithm called Bayesian optimization on our k-means classifier to do the hyperparameter tuning.
Now, generative adversarial network, known as GANs, are a system based on min-max strategy where 2 algorithms are confronted, and on the first hand we have algorithms that generate data, the generator. On the other hand, it generates discriminates between fake and the real data. This technique can offer a novel way to unlock additional information from a data set by generating a synthetic samples with appearance of real image. And on the right side, we can see the conjunctival melanoma time-lapse analysis. Next slide. And for the future, we are planning to increase the dataset in terms of size and metadata to analyze with different models and different time-lapse analysis. And we have three main questions to answer. So, can we scale augmented data source with PACS? Could we predict cancer before it shows? Does time series approach yield improvements? Next slide.
So in conclusion, for this type of body organ and its tumors, semantic image segmentation is better suited for the recognition of different types of conjunctival and corneal tumors because of the lack of a mass data set. In addition, fewer model layers decrease complexity when samples have no fixed geometric shapes and also a variety of gradient colors, and simulation with the time-lapse analysis boosts the performance of our model.
And also, I want to mention all the data and methods are open source in the GitLab already, and we agree to share our findings to other researchers group.
Thank you to our sponsors, organizers.
All right, go ahead. Yeah, and great, you have 2 minutes. So if anybody has any questions, judges, please chat or unmute yourself.
I have one question. Very nice work. Do you know whether the data set you used reflected eyes of a whole range of ethnicities and backgrounds?
No, we didn't have that type of metadata, unfortunately. But we are planning to add this type of metadata. And for example, sex, and some other type of information because they are essential to make some sense. I can see one more question in the chat. How much computation time does the 4 method cost?
Yeah, we have used
this is you.
Yeah, we have used a GPU to calculate. So for every epoch, for the poorest performance is around 5 to 7 seconds per epoch. But for the high performance, we have like 3 to 5 seconds per epoch. Any
Lucio? Yes, I can see one hand from Lucio.
Yeah.
I already asked my question. Sorry, I'm gonna lower my hand. Thank you.
Another question: could data augmentation have helped in this case to improve the performance? Unfortunately, we believe, in our opinion, data segmentation, from what we searched, has better results, but we didn't have the time to test augmentation. Don't know if this answers this question. And the next question is, as follow-up question, there is another type of melanoma in the eye known as uveal melanoma. Do you dataset contains type of uveal melanoma? No, unfortunately, no.
We have
but we are planning to expand the types of melanoma.
Yeah, just try this.
Sorry, we're out of time, so we need to move on to the next.
Thank you, everyone.
Team.
Thank you.
Yes, thank you. Everybody should be able to share their screen and unmute themselves now. I think we have it set that way. We don't have to worry about that. So the next team is, let me share my screen. Is team 1 called Team Ditto, AI-guided multi-omics data mapping on, sorry, I have some technical difficulties. AI-guided multi-omics data mapping on pathology whole slides images of glioblastoma. And I'm not going to list all the names there, and I'll let you introduce yourselves. And your time starts now. You can go ahead and start sharing and start presenting.
It says I cannot interrupt your screen sharing, Amy. Thanks. All right. So I will trust that you can see my slides unless someone says. Amy said our team title, and on the left are our team members, on the right are our team mentors. Sadly, a big chunk of our team had the GBS symposium today, but Thea and Rachi are in the chat to answer questions as we go. The big thing that we tackled was, in general, if you're making a cancer diagnosis or looking at prognosis, there are many modalities to use, genomics, epigenomics, transcriptomics, proteomics, and so on. But when you're sort of crushing up cells and putting them all in one tube and getting data, how do you know which cells were important? And if you've got a slide that's equivalent to those cells that you crushed up into the tube, how can you map the information from the big mess in the tube onto that slide? Doing this requires years of training and hours of annotation and interpretation for a human to do. And often it's just too much data for a human to consider. So what we tackled was integration of histopathologic and molecular data in an automated fashion, trying to connect what we're getting from omics data and image data through computer vision. So this is not in itself new. AI has been used to automate image analysis and is being investigated for lots of applications in pathology. However, most of those uses are supervised approach, where you've got annotations from experts and you're training the computer on thousands of expert annotations. Also, because it's a supervised approach, often those will end up overfit or hard to generalize. So we thought about it a little bit differently. Our big picture goal was to answer the question, by integrating computational biology and recent computer vision innovations, can we rapidly map omics data onto pathology images without supervision? So this is without a human giving us examples where this has been done correctly and then the computer learning from those examples. We wanted to do that in an unsupervised way to guide targeted therapeutic approaches. To do this, we use TCGA data, some of which is accessible through Ubright, and had some interesting computer vision discoveries along the way that we'll show you. The sort of pithy, trite thing that we're trying to do is trying to connect genotype and phenotype. We had two sort of goals for the hackathon. The first one, phrased as a hypothesis, is that an unsupervised method for images can annotate and label tumor cell subpopulations from a slide. And then as sort of a stretch goal, omics data can be used to guide and refine subpopulation labels. So to do this mini goal just for the hackathon that's coming out of our big picture goal, we were looking for a method that would take as inputs slides or sections of slides and a number of clusters that in the big picture you would get from your omics data. And to do that, we tested 5 different K-choosing methods and 7 clustering methods, looking for an algorithm that would accept as input the number of clusters, and as output, give us segmented, ideally labeled image. Sorry, one of my dogs is doing crazy. The details and somewhat novelty of this is that we skipped feature extraction. We clustered directly on the images, so we weren't requiring someone to tell us about the cells. We weren't using software that would try to pull out details about the cells. This was an entirely unsupervised approach. And we wanted our output to look like this. So taking the slide or section of the slide and labeling abnormal cells or areas of interest. This slide is mainly to convince you that we tried lots of things, lots of unsupervised clustering methods. And from 2 of those, we really got something interesting. So the first one, using mean shift clustering, these areas that are highlighted a little bit brighter blue than everything else, looked to the pathologist on our team like mitotic nuclei. We didn't have time, because this wasn't our plan even to go down this road. We didn't have time to do like a blind pathologist labeling to validate that. So as sort of a pseudo validation, we found a software package that claims to calculate mitotic index. And we tried to see what it labeled as mitotic nuclei. And we actually saw a good agreement between mean shift clustering directly on the image and this special package for labeling mitotic nuclei. So not a true validation, but certainly something interesting, but ultimately tangential to our goal. Second of all, we did achieve good image masking. So this is where a pathologist might say, these are the cells that we're now going to look at. In an automated and unsupervised fashion, we were able to pick out cells from background, essentially. And to do that, we use a pretty new method that we found on GitHub that was based on PyTorch. And this is a convolutional neural network that does not require label images to train and still gets out a, at the end, annotated image. And to tune that method, we used a new idea out of Google DeepMind research called population-based training. A little fun figure there. So we started with a raw image without labels. we were able to successfully mask that image without any pathologist intervention and without needing to do like a feature extraction step where we sort of pull out tabular data from the image. Now that we have that, and we've actually sort of achieved our hypothesis one, what do we do next? Well, now we want to actually label those cells in a detailed way. So to do that, now we had to bring back in feature extraction. We pulled out 11 different features based on size and shape of 630 cells, and then segmented that image, again, unsupervised. So we didn't need a human intervention to do this. Once we had that, remember I promised you something about omics here. So we've got clusters from the image, but now we want clusters from the genomic slide. And this is achieved by using biomarkers. These biomarkers and genes are naturally clustered together by participating in the same cellular pathways. So to find these biomarkers, we want to use multi-omics modalities. The idea is that a gene set enrichment tool could form clusters and that we can map those clusters to the images. So in the hackathon, with the timeframe provided, we focused on transcriptome data. And we did indeed get some hints on how we would link these clusters from the pathology slide over to the omics data. So during the hackathon, we succeeded in clustering cells on the pathology slide in an unsupervised fashion. And next, we're really trying to link that to the clusters that we got from the omics data. And that will require a little more help from a team of pathologists. But overall, the idea is we've got this labeled pathology image in an unsupervised fashion, which we think, again, is just the novelty here. That's telling us about phenotype. Next, once we know what the pathways are and sort of how prevalent they are based on pathologist information, we'll have the genotype. And the idea is that we could link those together and say, these are the cells that are actively participating in actin polymerization over on the right to guide targeted therapeutics. Oh, these are the acknowledgements, both the team members, mentors, organizing committee, and sponsors. And now questions, please.
Okay, we have just we have over 2 minutes left for questions. So good use of time. So there's already some questions in chat.
Gotcha. So I see, which omics do you plan to incorporate? Within the hackathon, we looked mainly at RNA-seq. This is a place where we're going to go grab a pathologist and say, how do we tell what the pathways these cells are participating in is? And so I will admit there's a little bit of, that was the big picture goal. So we're not there yet. But whatever we can get out of the TCGA database would be my short answer to your question. The next one's more of a statement, and yes, I agree that would be interesting.
I have a question.
Sure.
So currently you are assuming all the cells are cancer cells, is that correct?
No, not necessarily. So that was one of the things we think we could do, is that cell of interest. So tune the clustering method to instead pick out the tumor cells on the slides, and instead of picking out all cells on the slides.
Yeah, so because they do have some other cell types in the images, right? That could potentially like immune cells that infiltrated to the tumor regions. And when you try to link up with your RNA-seq from TCGA data, the enrichment potentially could be biased to more like a cancer cell signal.
Sure.
Have you kind of like think about how to address those issues?
So this was one of the discussions. If Thea is here, she might be able to say more, but the overall idea, actually I need to go back to the notes on that slide. Sorry.
I have about 50 seconds left. Just time.
Gotcha. So short answer is yes, we did have that discussion at length if we're almost out of time.
Any other questions? Well, if not, I know we're kind of behind schedule, so I think maybe we can just move on unless you have anything else to add. Got like 20 seconds left. All right. Well, thank you very much. I'm going to just let the next team start presenting, just to try to save time and not use up any more time than necessary. So it's team 8, yeah, the D team.
That's the D team. How to share the screen.
There's share screen button on the bottom you can pick what screen you want to present. There you go.
Presentation mode. Okay, so everyone can hear me?
Yep.
So we did a presentation on the impact of vitamin D-like compounds on oncogenes. And the members of my team were Reimer Saminsky, which is myself, Mohammed Salim, and Yue Song. So our project was to investigate how vitamin D-related compounds, Gemini 72 affects the gene expression of certain oncogenes and extracellular matrices in mouse prostate. So we used our data from this paper here, El Mate et al. Single cell analysis and Ravel single type specific responses to a vitamin D analog in prostate precancerous lesions. And here's the structure of vitamin D, and here's the structure of Gemini. It's very similar, but it's in the family. And what we hypothesize is that this extracellular matrix related degradation genes such as MMP would be downregulated since they play a role in cancer metastasis, and also the oncogenes, like those that promote cancer, would be downregulated by vitamin D, or this compound Gemini 72. So we looked at several types of genes. We looked at extracellular matrix genes such as fibronectin 1, fibrillin 2, and MMP 2 and 3, and we also use as immune markers. And the reason why we use immune markers because they tag the immune cells. This is used for immune cells to tag the cancer cells. So this is CXCL1, CXCL2, and CXCL5. And then we also look at cyclin-dependent kinases, and this one CDNK2A is an inhibitor. It actually suppresses cyclin-dependent kinases, while CDK4 is a main cyclin-dependent kinase. And finally, we looked at the oncogenes like melanocortin-1 receptor, ATM, which is serine threonine kinase, BAP1, which is the ubiquitizing enzyme, and finally MIT, which is microphthalmia-associated transcription factors. Materials and methods, we use two data, like the data sets, and one is the vehicle, which is sunflower oil, and the other one's the Gemini 72 compound. And what we did is we used a single cell RNA-seq analysis. We used a TISNI, which is this artificial intelligence program that clusters data based on the location. We also used a program RAR to analyze data. Over here, we have our TISNI plot, like a vehicle versus Gemini 72, and shows that already slight differences. And right now, we first go to our extracellular matrix, and we did the first of all fibronectin, like on the left is vehicle and right is Gemini. As you can see, there are slight differences, and like from what we're seeing here. So go on also to MMP2 and 3, and we see MMM2 with vehicle, and one to the right, I think there's more being expressed with MMP3 on the right. So it shows that, like not actually less expressed on the right. So it shows that like the vitamin D compounds are working, that the reducing expression of this MMP, which is metastasized. Then we go on immune markers like CXCL1, and we're also seeing a little bit of difference here, and also CXCL2, it's CXCL5. And we went to cyclin-dependent kinase, and so this is the inhibitor. There isn't much expressed, but from what we see here, that there is like a slight difference between the vehicle and Gemini 72 with the cyclin dependence inhibitor, and also with CDK4, there is actually there's, I think, less of it being expressed on the right from what I'm seeing here. And like you see, there's more here and less over there. And we also, finally, with the oncogenes, and it's hard to see, but we did see slight differences between the vehicle and Gemini 72, and the conclusion we have is that there are differences between the vehicle control and Gemini 72 treated. And we saw differences, particularly MMP2 and 3, and that this compound might be potential candidate for treatment of cancer. And if one future directions, maybe look at further genes on and maybe further investigation, because we use just the vehicle and vitamin D samples. Maybe in other futures, maybe to use more samples. So I think that's any questions?
Yeah, we have about 4 minutes for questions, so we got plenty of time. Questions from judges?
Is this the only vitamin D-like compound you tested? Because there are several, and they may differ. So it would have been nice to have an internal control with like vitamin D3 or any other vitamin D receptor agonist. That way you will know that what you're looking at is a vitamin D-like effect.
That's a very good point. Thank you for that suggestion. Any other questions?
RNA-seq analysis.
Okay. So I think we use the RNA-seq analysis to normalize the data.
What are next steps? What are future work that future areas of exploration and elaboration?
Sorry, is it okay if I get like Celine, if you can answer the question? Just how the next steps with the analyzing the data.
Do we follow the data set and then going through the normalization first, and then compare between the data set and all with the graphical?
Sorry, what I'm asking is there's two data sets, right? One is the vehicle, one is the treated one. And then when you present the t-SNE plot, you're presented like two different t-SNE. Have you tried to merge these two data sets?
No. I'm sorry. Yeah, I understand. No, we only work on the one data set. We didn't merge the other one. We only work on the sunflower.
What were some major challenges and issues that you faced, and how did you overcome them?
I would say one of the major challenges we had was, I think because we're learning R still, so we use this more as a learning experience, and what we're doing is knowing which programs to use, because R is very powerful. It's so many things you can look at, and also the genes, and it's just narrowing and finding which genes of interest. And we found that very kind of a daunting task at first. Just learning not only knowing the package to use, but know which genes, and so it's a bit of a challenge.
Thanks. Any final questions from the judges? All right, well, if not, then thank you for an interesting presentation, and we'll move on to the next presentation. And that is Team Pineam.
Excellent, thanks, Amy. All right, well, obviously, thanks for this opportunity for all the collaboration that the hackathon allowed. Our team is composed of Chris Willey, Kevin Lee, Isan Singapore, myself. I'm Josh Anderson, the technical director over at the UAB Kinome Core. This is all based on data we generated, and Isan really did all the heavy lifting in terms of actual coding. I'll try and breeze through this for time purposes, focusing on some of the more pertinent points. So our project was focusing on using some of our kinase signaling data that we had generated in glioblastoma, or GBM, using both basal signaling and ex vivo drug response data. We had previously profiled 21 glioblastoma xenoline tumors, each treated with 3 separate small molecule inhibitors of kinases, brigatinib, sitravatinib, and neratinib. We profiled these tumors on our high throughput kinase activity arrays that we call PAM chips. And the overall idea here was to use these kinase signatures that we generated to develop patterns and signatures for response to drugs that we could apply to new, incoming unprofiled patient samples. On these arrays, we generate a unique type of data as we were actually measuring the activity of the kinases in the lysate against over 190 immobilized targets. The data is collected kinetically over time. One of the benefits of measuring the activity is that we can spike in actual inhibitors on chip, or what we refer to as ex vivo profiling, and directly measure the effects of these drugs against, again, the hundreds of targets in our patient tumor lysates or cellular lysates and things like that. Of course, there are limitations to this. Currently, I manually have to go through the kinetic data curves and exclude data in a partially subjective manner. Unfortunately, there aren't any housekeeping kinases to correct or normalize to, and things such as sample handling time, temperature, and the amount of inert, such as fibrotic materials, can make it very difficult to normalize the data. In this project specifically, we wanted to identify basal and drug response patterns or signatures and apply these again in a predictive manner. More abstractly, of course, we want to use this data to combine it with other omic data in a user-friendly, like informatically useful manner. So as to our progress, just getting the data ready where it would sort of play nice with coding and properly describing it was a chore. But eventually, Ehsan worked some coding magic and developed this online tool. There's a link on the bottom right. Obviously, you can't click on that unless you're looking at the PowerPoint, but hopefully we'll have time to demo it later. He made the data displayable in an interactive manner where the individual peptides can be selected through drop-down menus shown here up at the top. Across tumor and drug treatments, displaying all available like exposure, cycle, and time formats, making a visually useful sort of encompassing overview. This should allow for the identification of problematic data, such as poor kinetic curves, bad cycles, and over or underexposure signal. I'll try and get through the rest of this pretty quickly so my teammates can correct all the things I'm butchering and possibly, again, show the action and show the tool in action if we have time. So the first coding task after formatting and importing the data was to take all the arrayed data, again, across over 194 peptides with multiple cycles from 21 tumors that we'd profiled. The data from the untreated with no drug, patient-derived glioblastoma neurospheres was used to identify the peptides and their correlations with other peptides across the chip, identifying peptides that travel together with high predictability, sort of allowing a reduction in dimensionality for visualization purposes. So the peptide correlations are filtered and mapped using a Kamoto-Kawai algorithms and Gaussian distributions to map the peptides onto a 3D terrain shown here. This is color visualized, essentially converting it where we can convert it into 2 dimensions. Yeah, if you have any questions about these formula, save them for the end. I think I understand these about as well as my dog understands how a carburetor works. So we were pleased to see that these terrain mappings of these peptide networks were able to identify and separate basal signatures, similar to what we'd seen and validated with other clustering methods previously. So for example, here we have 4 tumor signatures or terrains shown. And you can identify the 3 tumors that have pretty similar looking patterns, whereas the second one, X1066, has quite a different pattern displayed. Additionally, we were able to compare and visualize differences in varying phenotypic differences across a subset of these tumors. Here, we are actually comparing parental tumors to the same patient-derived tumors that were grown to develop resistance to the chemotherapeutic temozolomide. And as we'd quantified in a simpler, more supervised manner previously, we did see that there was a difference in how these resistance mechanisms developed, and we could visualize these differences in these kinome terrain mappings. We also highlight an important step in the ex vivo drug response testing, where we saw roughly 3 subtypes of basal signatures, as indicated by the red Roman numerals above. And this is really important as drug response modeling must really take into account the basal patterns. There's not necessarily a linear response to drug-based inhibition. We do hope in the future to find a way to normalize and account for this, but that was a bit much for 48 hours worth of work. We also had, well, here's an example of a single tumor showing drug response patterns. The basal signaling is shown up in the top left, and you can see with these green check boxes, we have responses to brigatinib and sitravatinib, which showed significant inhibition, whereas there's essentially no difference between the control and the neratinib treated displayed here. We also, this got cut off a little bit. We also have the ability to adjust the sigma, sort of where you can limit what is displayed. And hopefully Ehsan can show this at the end if I talk fast enough. For tumors with similar basal signatures, you can visually compare relative drug responsiveness. Here in the case of sitravatinib, we saw that this center tumor here, JX15, had a significant response to sitravatinib. Whereas the flanking tumors, JX12 and X1016, did not have a significant response, again, showing the usefulness of this tool. The interactivity of the tool allows for easy selection of tumors, specific peptides, and multiple other variables that we can add in. And additionally, the generated terrains, you can actually zoom in on these mappings. Here's just showing an expansion of this tiny little red spot here that shows a inhibition of IRS1 signaling in the drug-treated sample. And Isan, in about 2 slides, if you can bring up the tool live, or you guys can try it if you can access the link below. So we had some fun, got a few useful bits done, especially given the time frame. But of course, we'd like to continue sort of, obviously move forward with some automation, normalization, and pattern recognition as mentioned. Yeah, thanks everybody for putting this on. And if you have any questions, I guess pop them in chat or let us know here.
Yeah, right at 2 minutes, 2-minute warning. And I do see a question.
Oof.
Okay.
That was me. So how do you define ex vivo exposure? Do you have a range of doses, for example, of your kinase inhibitors? Usually these types of experiments are done on a dose response that reflects in vivo achievable concentrations. And also, how did you define a neurosphere? Those kinds of cultures often contains spheres of different sizes and shapes. And the closer they are to a patient-derived phenotype, the more heterogeneous they are. So you may want to consider that in your future development. Perhaps using organoids may be a better approach, because those can be made of a little bit more standardized size distribution, and are grown in collagen so that they're not going to float around.
Oh, absolutely. We actually are developing these 3D printed structures where we're integrating lots of like brain-derived or brain-related ECM proteins and printing like these high throughput arrays. For our dosing on our ex vivo testing, we've done some test dose responses. We're actually spiking these inhibitors into the lysates on the chip. So the dosing is, can be significantly different. We lose things such as cellular structure and sublocalization. So we sort of have to determine the dose in this specific setting as well. But no, we completely agree with developing the better models for testing these.
And really quickly, these were tested on xenograft tumors. So these are actually from animals. These were not, when we say ex vivo, what we mean is you basically harvest a tumor, lyse it, and then you interrogate the sample by spiking in, a vehicle versus inhibitor. So you're able to basically investigate the tumor out of the, live sample.
Thanks. Unfortunately, we're out of time, so I have to move on to the next team, and that is team 3, Team Sui.
So. I hope you can all see the screen.
Yeah, but I think I'm seeing your screen. You need to share your presentation screen, window.
Yeah, I think now it should be fine in this case.
Yeah, looks good.
So good morning all. Before I begin, I would like to thank the organizers of the hackathon for not only motivating us to learn the machine learning approaches, but also facilitating some very good collaborative effort. So as a team, we are a team of 4. We set out the aim to develop a machine learning model that can differentiate high-risk prostate cancer patients from low-risk prostate cancer patients. So in our team, we have Dr. Suri Varambali, who's an expert in prostate cancer research, Dr. George, who's been working on different machine models, and a very enthusiastic PhD student, Santosh. so who has been pulling a lot in terms of coding and all. And I've got good experience in handling the data, especially the high throughput sequencing data. So let me begin with the background. So as most of you are aware, the prostate cancer is one of the leading causes of the cancer in men in US as well as in the world. So it basically involves multiple stages. It starts as a small tumor which actually grows, and it first invades a small gland which is part of our immune system called as lymph node, and that's where it starts spreading to the other parts of the body, in this case, which we call it as a metastasis. So recently, a publication in European Urology, they have defined what they mean by a high-risk prostate cancer patients who are at risk to develop the metastasis and ultimately lead to death, and low-risk prostate cancer patients. So they have considered two main clinical features to determine this. One is called as Gleason score. So whenever a pathologist examines the sample from a patient, so they give a Gleason score which ranges from 6 to 10. So any patient with the Gleason score 7 or below, 6 or 7 basically, indicates that they do not have an aggressive tumor. So when the Gleason score is above 7, ranges between 8 to 10, that means the tumor that they have is very aggressive one. And then the other feature that they consider is nodal metastasis status. So as I mentioned in the previous slide, there's a small gland which is a part of our immune system that is nearby the prostate. So when the tumor grows and it grows out of the prostate, the first thing it reaches is actually the lymph node. So the N0 status indicate that there is no lymph node metastasis, there's no spreading to the lymph node. N1, N2, N3 are the status where there's progressively more spreading of the cancer cells to the lymph nodes surrounding the prostate cancer, surrounding the prostate organ in this case. So based on these two features, the paper I mentioned from the European Urology, they have classified the patients into a low risk. So patients with the Gleason score of 7 or less and with no lymph node metastasis, they consider them as a lower risk patients. The patients who have Gleason score less than 7 or equal to 7, but they have only one, that is 1 to 3 axial lymph node metastasis, they are considered as a moderate risk. And those patients who have greater than 7 Gleason score, and irrespective of the lymph node metastasis, they consider them as high risk. So with this information in our mind, we thought we will utilize the TCGA data, which is publicly available, and categorize the samples that is there in the TCGA. So TCGA provides multiple types of clinical features for each patient. It has information. So out of 498 prostate cancer, the patient information that is available in TCGA, when we applied these particular rules, we could identify 220 patients who are at low risk and 192 patients who are at high risk of developing the metastasis. So with this information in mind, we wanted to explore the RNA-seq data from the TCGA for these patients and apply the machine learning model approaches. So the first stage of our project was the data preparation. So as you all know, TCGA provides the level 3 data, which is not controlled. So one can download the raw read counts for each of the raw read counts for each of the patients. And we performed the differential expression analysis to identify genes that are up or down regulated in high risk patients compared to low risk patients. One of the main reasons why we do this is because TCGA provides information for about more than 60,000 genes, coding, non-coding genes and all. So one cannot begin with that level of, again, we thought that we will focus mainly on the genes that are differentially expressed in high-risk patients. So we obtained, in total, we got about 749 genes that were differentially expressed. There were 481 genes that were upregulated and 268 genes that were downregulated in high-risk patients compared to low-risk patients. So once we got this, we downloaded the gene expression values. Currently, TCGA provides something called FPKM values, which is again normalized gene expression values for 60,000 genes. So only for these 749 genes, we obtained the data matrix. So we had the sample information as well as the expression values for each of these 749 genes, and this actually formed our initial data. So the second stage of our project was the feature selection. So even 749 is, we thought it's a pretty big number, and we wanted to identify what are the real important features among or genes among these 749 genes, because differential expression works in a analysis works in a different way. So we actually performed something called as recursive feature elimination analysis. So it's a machine learning approach where we divide our total data into a training set and test set. And then in this case, we have applied random forest classifier, which basically identifies the subset of features or genes which can more accurately distinguish high risk and low risk samples, all using the gene expression values that we have in this case. So when we applied the recursive feature elimination method using sklearn modules, so we got about 347 genes out of 749 genes, which were optimal. And when
2-minute warning.
Pardon?
Sorry, 2-minute warning. You have 2 minutes left.
Okay. So in that case, so with the test data, we found out when we applied these 47 genes, we got about 76% accuracy. So this is what we used for the for testing our machine learning models. So in total, we have now for the final phase of our project, that is to apply machine learning models, we have data matrix of 412 samples, as I mentioned, about high risk and low risk sample, and gene expression information for 347 genes. So as with this hackathon, we had a very good session with the PyCaret to understand PyCaret and its utility. So we used that information, knowledge. And for this particular analysis, we used the PyCaret and we did it in the Chia system. Server. So basically,
have 1 minute left. It'd be nice to leave some time for questions if you have any, or just maybe.
This is my last one. Slide in this case. So I'll be basically we have 3 different sets. So data was divided into 3 sets. That is training set, test set, and unseen set. We applied supervised machine learning models that is available in PyCaret, and we selected 7 models which showed accuracy of minimum of 70%. And these 7 models were actually tuned further to improve its performance, and then finally applied on unseen data, which were separated initially. So among them, AdaBoost classifier was the one which showed the highest accuracy and a good AUC score for our data. And so this till that we have done it, and we want to implement this on multiple independent prostate cancer data sets and want to validate our models accuracy further. And also we want to explore on the survival information of these 347 genes to narrow it down on small gene set, which can help clinicians or physicians to identify the high risk patients in the case where the clinical features are not available. By using
Thanks. Sorry, we're out of time. I know we don't have any time for questions, so sorry about that. All right, move on. And the next team is is team 9, Team Tokyo.
I will share my screen. Can you see my screen?
Yes.
Okay. Hello everyone. Our team consists of 4 people from University of Tokyo. Our research project is exploring multi-drug response with genotypical and chemical features under a novel deep learning architecture. Drug resistance is a serious problem in cancer therapy. Tumors often exhibit drug resistance over the course of the treatment. Consequently, patients who initially respond to therapy regress as their cancer either adapt to overcome the drug or an existing resistant subclone repopulates the tumor. Mechanisms of resistance include feedback selecting for upregulated expression of the target protein, altered DNA damage response, modification to enzymes involved in drug metabolism, intratumoral heterogeneity, and so on. Drug combinations are regarded as crucial for addressing this issue of drug resistance and preventing recurrence caused by remaining cancer cells. Synergistic combination can also reduce toxicity by allowing for lower doses of either drug. By enabling reduced doses, drug combinations can further increase the feasibility of drug repositioning by increasing the potency of compounds which has relatively high toxicity. Therefore, multi-drug therapy against cancer is recently attracting attention. Let me move on to the next slide. Oh, sorry. Okay. The degree of synergy is experimentally measured by testing 2 drugs as a particular number of different half-log dilution concentrations, which gives an n times n dose response matrix and quantified according to a theoretical model such as lower additivity model. However, such trial and error combination design has limited applicability in the clinic level due to time constraints and potential hazardous exposure to toxic combinations without improving efficacy. One solution for this matter is to use computational methods to explore optimal drug combinations for a patient. Many computational methods have been developed to predict anti-cancer drug combination synergy based on a variety of gene expression, chemical feature of drugs. Based on these backgrounds, yes?
Okay. Well.
Okay. You can continue, yes.
Okay, I will continue. In this project, we have 2 main ideas. Firstly, we want to explore gene expression effect on multi-drug response prediction. And secondly, we want to explore if using chemical features based on graph convolutional network can replace the traditional chemical features, where the traditional chemical features include counts of extend connectively fingerprint with a radius of 6 and predefined cyclical chemical properties and the binary toxical features, where toxical features are substructures which are known to be toxic. Next slide. We develop deep synergy model as baseline model. Deep synergy model is simple deep learning model like this graph. The model includes chemical features of drug A and drug B and gene expression of cell line and finally predicts the synergy score. To explore gene expression effect on multi-drug response prediction, we first prepared 2 data sets for training and predicting. The first one is attached to deep synergy. This data set consists of 4,389 chemical features of drug A and same chemical features of drug B and 3,984 gene expression of cell line. And this data set covers 38 drugs and 39 cancer cell lines. And secondly, we replaced the gene expression of deep synergy data with that of deep drug response data. We didn't use cell line which does not contain in deep drug response. So we used 8 cell lines. In this data set, it consists the same chemical features of drug A and drug B, and 18,400 gene expression of deep drug response data. And next, we join deep synergy data and gene expression of deep drug response data, not replace a gene expression of cell line which does not contain in this deep drug response was filled with 0. This data set is consists of the same chemical features of drug A and drug B we introduced previously, and 3,984 genes expression of deep synergy data and 18,400 gene expression of deep drug response data. In the other hand, we also want to explore if using chemical features based on the graph convolutional network can replace the traditional chemical features. So we developed a chemical information extraction model based on graph convolutional network. The pipeline of the GCN model is shown in the graph. First, we include chemical complex smile representation, like in the left part of the graph. Then we extract molecular information such as atom, bond, and structure as input of GCN. And finally, we obtain a 128 dimensional vector for each molecule as the chemical information. And we integrate GCN chemical feature information to the prepared data in 2 ways. In the first way, we combine joint deep synergy data, which we used in the previous part, with the GCN chemical information. The data set contains the same features of drug A and drug B with 100 chemical features we extract by GCN. And in the second way, we combine joint deep synergy data without drug chemical information with GCN chemical information. This data set contains 128 chemical features of drug A and drug B separately, which is extracted by GCN, and other feature is the same as the first data set. Well, unfortunately, we made the mistake during the extracting GCN chemical information. We extracted GCN chemical information by initializing the model parameters with random number rather than the trained model parameters. So the GCN chemical information extracted in our experiment becomes a random noise. Well, we finally obtained the result in different data sets shown in the table. We calculate RMSE with different data sets to evaluate the performance. Comparing joint deep synergy data with deep synergy data and changed deep synergy data, we find that more genes using as gene expression feature helps a little in exploring multi-drug response. For GCN chemical information, we will try the correct model as a future work, but in the result of this experiment, we can see the importance of chemical feature by comparing joint deep synergy data results and joint deep synergy data results drug chemical information result.
You have just under 1 minute, just under 2 minutes left.
Okay. To summarize our project, in this Hackathon project, we built a test pipeline for investigating different features used for the binary drug synergy score prediction based on the deep synergy framework. And we evaluate the effect of number of genes used for the gene expression and propose a prototype of using GCN embedding features for the chemical information representation. That's all, thank you. And any questions?
Thanks. You have about a minute left, and there are a couple questions from Lucio. If you could just take a look at those in the chat and respond to those. Yeah, everybody try to look at them before the end so that you can think about it before it's question time.
Okay, thank you.
So first question, do you know whether drug sensitivity was assessed under 2D or 3D conditions? The latter is much more representative.
Actually, I'm not sure about the the dimension, so I will check later. Sorry. And
Okay,
the
second
next
question, yeah. Can you predict synergistic toxicity to the patient as opposed to the cancer cells? Many potentially attractive drug combinations cannot be used in vivo because of additive or synergistic toxicity.
That's an important point. So there's a difference from in vitro assay and in vivo assay. So in vitro assay result is not always applicable to in vivo assay. So we have to test in vivo respectively, I think. You call a mistake, but it also makes for an interesting baseline for the test. Oh, Liu-kun, could you answer this?
Yep. Sorry, we're out of time. And
Is that a question?
Okay.
Sorry about that. Yeah, it's just a comment that maybe something interesting could come from that too. A lot of comes from so-called mistakes. Okay, so the next team is team 12, colorectal coders.
All right, I think that's up to me. All right, can everyone see? Okay. Okay. So hey everyone, my name is Justin. So I'm going to talk to you guys about today our project, which was detecting colorectal cancer in CT images. So I'll preface with we are all of my team members were kind of new to the field. So our presentation certainly won't be as technical as the previous presentations, but we'll get started. Okay, so yeah, we set on a mission to see if we could detect colorectal cancer in CT images. Of course, this has been done before and doctors can do it by themselves. Our team name, we decided to call ourselves the colorectal coders because we thought that was cool. It's not letting me mix. There we go. Okay, so here we are. My name is Justin. I'm a PhD student in chemical engineering at Auburn University. Here's Peter Abraham. He's an undergraduate student in the same lab that I work in, also at Auburn University. Ife Alula is also an Auburn University student and getting his PhD. And he's getting his PhD in nutrition. And then Asma, she's getting her PhD in public health and community medicine, and she is attending Cairo University in Egypt. All right, so here we go. Oh, I guess a little bit of the background of the timeline. So Peter and I came together and thought it would be cool. We kind of heard about this kind of late. So we started on the second day on the first day of the hackathon, planning everything and putting everything together. So our work is kind of behind on, I guess, where we could have been. And then later on, we added additional members as we went through the 2 days of the hackathon. So, all right. So why colorectal cancer? So colorectal cancer is any cancer that affects the colon or the rectum. It's the third most common cancer in men and women, and it's estimated that over 53,000 people died in 2020 from colorectal cancer. And I actually was so new to the field, I didn't even know what the colon was, and I found out that it was the large intestine. So colorectal cancer can start off as polyps in the colon. And I put an image here just because I wanted to see what that looked like. So I thought it would be nice to share with everyone else of just one of those polyps starting and ending over time as an invasive cancer. So it's very treatable when discovered early, which brings me to my next point, which is detecting colorectal cancer. So there are many tests that can be performed to detect colorectal cancer, and they're all listed here. I'm going to specifically pinpoint CT scans and colonoscopies. So CT scan or a colonoscopy can be used for early detection, but a CT scan is less invasive. So I put a little image here of the perks of CT colonography versus colonoscopy. And here are some CT images of the colorectal region of the body. So CT scans can be taken before just as a general image, not specifically just to detect colorectal cancer. CT images can be taken of before and during treatment after detecting colorectal cancer to look at the size of the colorectal cancer over time with treatment. All right, moving along. So why did we decide on colorectal cancer imaging and what were our goals? So colorectal cancer, so we knew that we wanted to learn how to do image analysis with Python because there are some ideas that Peter and I had that we wanted to learn image analysis such that we could apply it to things that maybe we will do in the lab in the future. So why colorectal cancer? So Peter actually works on colorectal cancer in our research lab. So we figured it would be a good fit. So that took us to our next point, which was to do a Google search on colorectal cancer, detecting colorectal cancer with Python imaging, basically. And we could not find anything that gave us like a clear answer or any like instructional page. So I put a Google search. There is no colorectal learning pages on how to do this. There are lots of papers, but there are no instructional articles. So our goal was to create a code that is easy to follow, it's reproducible and easy to teach and to learn. And it's also shareable through instructional web pages. Another one of our goals was to learn Python. So we were all new to Python. So we absolutely had to learn Python. Another one of our goals was to detect colorectal cancer in CT images. So we came up with a general plan for our code. So we would input a CT image of any colorectal CT image. And then we would use machine learning to compare this image with previous cancerous and non-cancerous images. And also, if available, compare previous patient images to this original input image. That was step number 2. Step number 3 was to identify regions that have the potential to be cancerous. We didn't know how we were going to do that, but we knew that was something we wanted to happen. We later learned on how we wanted to do that. Step number 4 was to produce an image that highlighted this potentially cancerous region. So this was our plan overall, what we wanted to happen whenever you put in an image. We wanted you to get out an image that could detect this potentially cancerous region. So thanks to the internet, we learned a lot from just a few snippets of web pages that we used. We learned how to use Python, what machine learning even was, what exactly it means for machine learning, templates for machine learning, which brought us to our plan, which was to use support vector machine learning. So we went through the different types of machine learning methods, and we felt that the support vector machine learning was the best method to use for our purpose. So we would have 2 portions to the code, a training portion and then a testing and evaluation portion. So for the training portion, we would feed it in annotated CT images from this website here. And it would learn how to do it. And then you could put in later on, after it's trained itself, you could put in an image and then it would be able to export an image that has a highlighted region of potentially cancerous regions. So the 2 parts to our code, there was the training and then, sorry, it's lost my mind, the training and the testing and evaluation section. So the training section, here's how we ideally wanted our code to work. So we would import the image, we would convert that image to an array such that the code could actually see the image itself. Then we would use machine learning to find parts of the body in that image so it could detect the small intestine, the colon, the rectum, and more. Then we would have the code ask the user to identify if it correctly identified a certain part of the body. For example, if the small intestine were in the image, it could locate the small intestine and ask the user, is this the small intestine? And then based on what the user would say, either yes or no, the code would save this data to a file to remember it. Then it would compare organs with previously saved organs to detect differences. So if we had a small intestine that was healthy, if we had a small intestine that had a polyp in it, it would compare the 2 and see, hey, there's this little polyp here. Then it would ask the user if the detected differences in those images are cancerous regions. And based on what the user would reply with, yes or no, it would save the data to a file to remember this, such that it could learn. So Next is the actual testing and evaluation part. So after we've done the training, this is the code that would be used to detect the actual cancer. So we would import the image and convert the image to an array again. We would use machine learning to find parts of the body again. But then we would use machine learning to detect differences in healthy organs versus the original image. We would use machine learning to find the area that is unlikely a healthy region or a region for concern, so a cancerous region. And then it would create a highlighted area by coloring this region. And then it would export an image that would have this highlighted region on the image.
You have about a minute and a half left.
All right, so overall, we didn't get to create code that actually did this, but we did learn a lot. We learned how to use Python. We learned about colorectal cancer. We planned as if we were to have enough time to do this. So yeah, that is our project and detecting colorectal cancer in CT images using Python. All right, any questions? I'll look in the chat.
Yeah, questions? What might you do next? So this is a great exercise in learning and coming from not doing this much. What might be the next thing you do?
So the next thing we started to create the code. The next thing would to actually finish and create the code. So because we're still all new to this, we still don't really know what exactly we don't know. So I guess the next step would to be create the code, go through any bugs, see if it works, change whatever we need to change. And if it doesn't work, and then hopefully we could use hopefully we could actually input an image and see if it detects the cancer.
And there's just a quick comment from Lucio that it's very preliminary, promising. CT colonoscopy has gotten a lot of attention. It's attractive. It's not invasive. Who wants a colonoscopy, but not widely adapted because of lower sensitivity. My alarm. So time to move on. So
I'll stop saying.
Yeah, thank you very much. Next team is I gotta look at my list is team 4, phenome miner, and just while we're making that transition, just to remind everybody that for the MC BIOS award, that you need to become an MC BIOS. One person needs to be an MC BIOS member in order for you to be eligible for that award, Interdisciplinary Team Science Award. Just a quick, that was a word from our sponsor. All right, so I think the next team is ready, so take it away.
Good morning, everyone. I'm Marat Mishra from Team Phenome Miner, and our team members are Nilesh Kumar, who's a PhD trainee. I'm a PhD candidate, and Dr. Lu, who's a postdoc in our Department of Biology. And we are going to talk about a natural language processing system for retrieving information about cancer phenotypes from published articles. So the main problem that exists till now is that however, there are several databases related to human phenotype ontologies. Still, there are quite a lot of gaps are there, and specifically in human phenotype ontologies, it has approximately 324 phenotype terms related to 852 genes in cancer. Though it is widely accepted, but it's not complete, because what we found that a huge number of genes which are characterized in cancer, they are not annotated or they are not assigned to any ontologies till date. And when you look over just a local search, that in nanostring pan cancer atlas, there are approximately 1454 genes which are associated with any kind of tumor related to cancer. While human phenotype ontologies, it had only 852 genes associated with any cancer related phenotype. And this coverage is only 18% of the known cancer gene. So there's quite a huge gap, which we are planning or we are moving forward to try to accomplish, fill that gap a little bit. So the next problem is that when you try to study or start studying any cancer, you look for literature. So the first step is to go to PubMed, and then you look for that cancer. And to read that whole article, it takes a lot of time. We have to screen the title, select a particular species, then read the abstract, then read the full paper. It takes quite a long time. And we are trying to solve that issue just by using a machine learning model or NLP model so that we can characterize or we can look for the gene of interest or phenotype of interest, and then in a short period of time.
So as we already mentioned in the previous paper, enormous amount of research article is already available in the internet, and most of them are indexed in PubMed. And there are almost 700,000 papers available open source for text mining in PubMed. Reading and parsing these papers for extracting phenotype information is a very difficult task. And if we are going, if we think about like going through all these papers, it would take like a year to understand how genes are associated with phenotype. So to mitigate this problem, we need some machine learning help to make it possible. So there are three popularly known machine learning model which are very efficiently able to solve NLP problems, namely GPT-2 and BERT and Transformer XL. Apart from these three models, there are several other models as well. But BioBERT is a BERT-based model which has been trained on biomedical articles, so it makes sense to use of this model to solve our problem. So in our approach, what we did is try to learn embedding from HPO definitions and going through the paper line by line and trying to make sense which line is similar to which HPO definition, so that we can annotate those lines with HPO IDs. So for our purpose, we need two things as far as data set is concerned. We need all the definitions from HPO database. For example, here we have lung cell, large cell lung carcinoma. HPO ID is there and the definition is below. So we want to first of all convert this definition into embedding using NLP model. And then we need papers. So we need to understand this paper paragraph by paragraph, and using line embeddings, we can figure out which is the most relevant definition available in HPO database. So our pipeline starts with SBERT, which is a modified version of BERT, especially designed for sentence learning. And in the background, the context on which this model was pre-trained is a BioBERT, which itself is consisting articles from PubMed and PMC. Then, as I said earlier, then we need all the definitions, and we fed these definitions into SBERT, then we get our embeddings. So this is the most essential part, and we need to just do this task once. After that, using PubMed API or downloaded XML document, we get our article. By candidate article, I mean that we will process only those articles which has at least one human gene and animal of interest, which is human for our pipeline. Once candidate article is filter is passed, then we using beautiful SOAP, we break down the whole document, whole body of the research article into paragraphs. And further, each paragraph is now further break down into sentences using NLTK sentence tokenizer. After that, we have a list of embeddings from phenotype HPO or regarding phenotypes and each candidate sentences. So using these two embeddings, one by comparing all the embeddings from HPO and comparing each sentence embedding, we can feed this into cosine similarity and we can get similarity score. So based on all the similarity search, top candidates would be chosen and accordingly HPO ID would be assigned. This is an example. So for regarding this paper, we just need to give address of the data set And it could be, the method is very flexible. It could be offline or online. If online, then we just need to give PMC ID. And then first of all, it gives us a meta information about the paper, and then internal assessment is here. So this, for example, in this sentence, the most interesting thing is here is that the sentence doesn't contain any gene, but its context is very similar to the definition of large cell lung carcinoma. That is why it gets similarity score of 92%. And the reason, and after that, we can make a correlation between gene mentioned later or before in the paragraph, and make association with HPIDs. So this is the more human readable tabulated format of that output. So we have list of genes, HPIDs, name, and their score. Later on, we can apply some filter and choose the most appropriate one. Regarding quality assessment, PubMed, so what we did, we processed about more than 1,000 PubMed articles and found total gene like 3,000 genes. And regarding those genes, we were able to predict 227 HPIDs. Further, Dr. Liu will continue.
So here, when you see that based on our predictions, we found that most of our identified phenotypes or HPOs and genes, they are associated with higher depth. So the depth number means that suppose we start with neoplasm. So neoplasm is now just the one depth, and then you go in hierarchical fashion, you go towards the more precise information or more this precise ontology. Suppose you go to depth number 7, then you'll get more precise information. Then you go to depth number 6. Similarly, the precision of ontology increases in the higher depths. So this will improve our analysis. That suppose not in lung cancer, when you go to a small cell lung cancer, a small cell lung cancer, then it's quite precise.
You have about a minute left.
Okay. So this is a comparison of 5 minor with the HPO and another strain database. Till now, HPO identified 852 cancer-related genes, which represents 18% genes of large-stream pan cancer, while 5 minors identified 3,013 cancer-related genes, which represents 55% genes of large-stream pan cancer. So, next slide. Next slide. Okay, so in the future, through some modification, 5 minors can be extended from cancer to rural disease. In addition, the workflow developed here can also be used for plant or yeast. And last, 5 minor also can be involved with more cancer-related research paper published. 5 minor will be included in more genes and HPOs. So thank you for listening. And questions?
Unfortunately, we don't really have time for questions. I guess there were some quick comments that genes with more than one phenotype, and the cluster of genes by shared phenotypes expand the correlation with other genes with phenotypes. These genes work together. All right, so thanks for the presentation. I'm sorry we don't have more time for questions. And the next presentation is Team 11, 2I cancer. I'm probably not pronouncing that right. So Team 11.
Hello.
Yep, I can see your screen. But yeah, you might want to.
I share my screen.
Yep.
Oh, so yeah. Can I see my screen?
Yep.
Okay. Our team members are from Tuskegee University. So including me, Xiaochang, and Dr. Stephen Suzuki and Dr. Fan Wu. We all play important roles in this team. Dr. Fan Wu organized this team, and Dr. Suzuki point a direction, the research direction, and provide valuable thoughts for our team. So I just do some coding. So our project is, title is Breast Cancer Risk Prediction with AI using clinical factors and mamographic breast density. So the breast cancer risk assessment is very important for identifying women who may benefit from more intensive breast cancer surveillance. So we have two goals. The first goal is build a breast cancer risk estimation model with AI and using clinical factors and mamographic breast density. So another important the goal for us is to investigate the racial disparity in breast cancer risk estimation. So we use a public data set, this breast cancer risk estimation data set with about 2 million screen monograms from women included in the breast cancer surveillance consortium can be downloaded from this website, their website. So in this data set, this data set includes cancer registry and pathology data, which are linked to monographic graph data and incident breast cancer, including invasive and ductal carcinoma. Within 1 year following the index screen monogram was assessed. So here are the factors including this data set, including some clinical factors and age group. So the breast density is an important factor has been included in this data set. Why we choose this data set? Because we want to do the this data set has a race information in it. So we want to investigate the health disparity issues in the cancer research. So has a white and Asian and Black and Native American and other mixed races, and has a lot of unknown. So in the study, I just took the unknown population out of the data set. And other clinical factors, BMI, age, so age at first birth, and a number of first degree relatives with breast cancer and other factors. So some factors are numeric or numeric data, but in this data set, they convert all the data to category data. So they just provide this data set with private category data. So has more clinical factors. There are 12 factors in total, and has the diagnosis of invasive breast cancer within 1 year of index screen mammogram and the diagnosis of invasive or ductal carcinoma, breast cancer within 1 year of index screen mammogram. So these two items or the attributes are the target we are going to predict. So we use 12 factors to predict the value of these two items, invasive and the cancer. So we I'm not using the we're not using the training label. We just split the data set to training and testing side by ourselves. So here's the risk profile of the cohort. So we can see the most population in this cohort is white. So just around 100,000 white women and about 10,000, around 10,000 Asian and Black women, and the less than 10,000 Native American and other races. So the method, we use 12 factors to make a prediction and all the data category data. So we choose the random forest because random forest is good to deal with category data. So we split the data into 80% and 80% for training and 20% data for testing. So here is the result. So it's the accuracy of predicting invasive or ductal carcinoma, breast cancer within 1 year of index screen mammogram using random forest. So all the values in this table or percentage. So we trained we did in an experiment. We trained first. We trained a single prediction model, use all the data of all the races. Then test the model on the population of different race, the white and Asian and the Black, Native American and other. And the second experiment, we trained a race-specific prediction model. So we just pull the data for each race out and train a specific race-specific model and test in the data of this for this race. For example, using the 80% white the data of white women to train the prediction model, then test on the other 20% of the data of the for the white women. So we okay.
You have about a minute and a half left. Sorry, it's running late.
Okay. And here is the accuracy of the predicting invasive breast cancer within 1 year of index screen mammogram using random forest. I did use the same method to train a single prediction model and a race-specific prediction model. The interesting thing is the white women has a more largest population in this data set, but the model trained with the largest population get the lowest accuracy prediction accuracy. So that's an interesting way we found in this study. That's what is not we expected before. So the conclusion, so the breast cancer risk could be estimated effectively by using random forest and the clinical factors and the mammographic breast density. The prediction models achieve the lowest prediction accuracy rate for the race with the largest population in the cohort. So we would like to acknowledge the BCIC release the data to the public. And our thoughts. So this project brought our interdisciplinary team together. So that's the important fruit of this program for us. So our team have already pursued the initial funding opportunity under Dr. Sudoke's leadership. That's the so that's a benefit of the program to us. Thank you.
Yeah, thanks. Sorry, we're out of time. Just a quick question is, you talked a lot about the ethnicity or race. Did you have information about the age? What was the age range?
We didn't, in this study just 2 days, we didn't get time to do that. But for the future, we will look into the different things, the age and other.
Right, great. And sorry we're running so far. It's going to be recorded, especially for the judges, if they need to watch something later, they need to get to another meeting. So the next team to present will be team 7, team pathology AI.
Okay, I'll try to share my screen. Can you give me permission, please?
Try it again. You might have...
I see it now. Thank you. Okay.
Yeah, just presentation mode, and then
yeah. Sorry, trying to get there. This thing is in the way. Okay.
Perfect.
Sorry, Zoom is giving me some issues. Yeah, I'm pressing back. Okay, there we go. Sorry about that. Okay, so I'll go ahead and start this presentation just to give you a little background about. Sorry, I must have put it in some automatic start. I'm going to have to just not have this in presentation mode, unfortunately. I'm sorry about that. PowerPoint is not ideal. Okay. The field we're working in is triple negative breast cancer, which makes up a minority of cases of breast cancer, but it has fewer treatment options. But immunotherapy has shown good promise in this field, and it also has the advantage that it has a lower risk of relapse. And as of 2019, a checkpoint inhibitor for immunotherapy in this was approved. So this is a clinically relevant field. One factor in having tumors that might respond is the tumor mutation burden. This is kind of counterintuitive because normally you think of more mutant coding mutations get expressed by MHC molecules. So it actually shows more neoantigens to your immune cells. So it gives a better target for immune therapy. So basically, if we can know that this, but the current method, or the ideal method, is whole exome sequencing, which is slow and expensive. So we were hoping to look at slides, standard pathology slides of these cancers, and then to predict. We kind of, our team divided up and kind of did a bake-off, and we tried a few methods. The one that showed the most promise is called CLAM. It's an existing method that we found in the literature. I can also kind of share my experience of what I did. I used the famous Keras and TensorFlow libraries in Python that open up Google's previously trained networks. And I actually used a component of what the CLAM method does, the ResNet 50 model trained on the ImageNet dataset. And then those libraries give you a way that you can substitute your own high level logic there and sort of train only that part. So you don't have to relearn all the low level image processing. So if anyone's interested in seeing that code, it's in the GitLab in case you want to try to play around with your own models. It was a little too simple and did not have great accuracy, but I'll show why the CLAM method expands on that and seems to do really well with these whole slide images. Basically, it takes a whole slide, which is a composite of many images on the slide. It divides them up, it pre-processes into HSV space. So it's an initial pre-processing there. It puts it through the ResNet 50 model. Then it puts it into, I believe, 1024 dimensional vectors, which conveniently you can fit multiple sample vectors into a GPU. So that's how they're able to get good performance there. And then they start with their gated logic, where they're basically dividing up the slide into many different squares and trying to figure out which ones are the most informative or the least informative, so they can kind of learn from there. As opposed to maybe a traditional image analysis problem where you're looking at the whole image at once. They're trying to figure out what are the actual relevant, maybe cancer cells as compared to other immune cells or other things like that. This is just some more images that came from their paper as cited here. Again, the idea is they're breaking up the slide into many squares and they're trying to figure out which ones are the most informative. So if you're interested, I would read that paper. It's very well written. For the data set we used, we were able to find a breast cancer data set in the Cancer Genome Atlas. We had 101 patients. Some of those patients had multiple slides, which the model can account for. And we trained on 80% and tested on 20%. We didn't have a separate validation set in this case. And this was the TCGA, so they had done whole exome sequencing, so we knew what the mutation burden is. And we discretized into median values, so lower 50%, higher 50%. For results, the results were actually pretty promising. We did five runs. We had some with higher, some with a little lower, but the average receiver operator characteristic area under the curve was about 0.7. So it seemed to be training an informative model. It would be interesting to see in the future, could we optimize it a little more and maybe get better results. But again, I think it's pretty impressive that just by looking at the images that it is able to get some insight into the mutation burden. I would be interested maybe if a pathologist has a perspective on that. Maybe they're so mutated they have some obvious morphological change or something. So to conclude, we went into a clinically relevant sphere and we applied an existing method to a new data set and seemed to get promising results. So again, to walk you through, the idea would be that a pathologist can put their slides through this algorithm and have an idea of should they order immunotherapy or not for this triple negative breast cancer. This is our team, and I'd like to thank especially our mentor, Professor Juan Liu, and our team leader, Yuki Chen, who is a physician who's interested in this. So thank you for your time, and I'm happy to answer any questions, although I know we lost some time at the beginning. I'd like to not thank Microsoft. I always have interesting experiences with them.
Yeah, thanks. Just a quick comment from Lucio. That's an interesting idea. It needs more additional validation. And yeah, I mean,
yes, definitely more data points. And I think the gold standard I saw written in the CLAM paper was if you can validate it on slides that were taken at another institution too, you can kind of control for how maybe different processing standards and things like that.
Yeah, I have a quick question. And you might have covered this. Were you looking at immunotherapy as a separate parameter or immunotherapy in combination with other therapies such as chemotherapy?
I believe it would be in combination. The paper I was seeing, it sounds like they do the immunotherapy, which I think is it's like an immune checkpoint inhibitor, but I believe they also do some DNA damage and other standard chemotherapies. That's my understanding at least.
It's FDA approved in combination with nab-paclitaxel.
Okay, great.
So if you're gonna, that is the indication. And that's what tumor mutational burden is used for at this time. Also, you may want to expand into, there are a number of other indications in triple negative breast cancer and in other cancers, actually, for use of immunotherapy plus chemo. And these are MSI, microsatellite instability, which does lend itself to machine learning because it can be detected indirectly immunohistochemically. And a little more complicated is single-strand repair defects. If you can see those from an image, I would be highly impressed. Nice work.
Thank you.
All right, great, thank you. Any other final questions?
I have a kind of question and comment. This is Rakjeje. I'm one of the neuropathologists here. So TCGA has some issues with the slides they have there. There are two sets of tissues, one frozen and one formalin fixed. Frozens are usually poor quality. And the other issue there is the slides are not scanned at the same resolution. So have you looked into those two issues?
No, not specifically, but that is a good specific question that could be looked at. I also may reference the paper we cited. I'd be interested to see, because they talk about using some public data sets. I'm not sure, I can't remember if they were all TCGA, but it would be interesting to see if they looked into that.
Because those two factors may bias your results in the end.
Oh, yes. Yes, that's why I think maybe using locally stored samples would be a great way to validate.
Thank you.
Thank you.
And thanks for highlighting that issue that you would think that the slide images are all the same and homogeneous, but yeah, something you have to worry about, something like scanning resolution. And yeah, so that's lack of interoperability, right? Okay, so thank you. Next presentation is Team 5, Birth-Seq AI.
All right, let's see. I hope it goes in the presentation. Excellent. Is it showing okay for everyone?
Looks good.
Absolutely. Excellent. Thank you, Amy. Hello, everyone. I'm Brandon Wilk. I'm the team leader for Team 5. Particularly, we're focusing around AI, around pregnancy-related conditions. Along with my fellow collaborators, Capri, Sharita, and Kristen, and Dr. Lizworthy. So we decided to focus on, for this AI competition, looking at the development of an AI-based approach to better understand a cancer called cholangiomas of the placenta and the heterogeneity related to that tissue. The reason this became so interesting to us is that there is a huge racial as well as regional disparity, particularly around pregnancy-related outcomes. And not so much to highlight the exact numbers here, but more so just to indicate these disparities are a consistent problem even to this day. A cholangioma is a tumor of the placenta, typically from vascular tissue, as you can see in the image in the bottom right. There's quite a large tumor there. These happen to also be associated with other conditions like preeclampsia, as well as fetal conditions and associated with negative outcomes from pregnancy, not just for the fetus, but also for the mother. And it also has known increased risks of other forms of cancer as well, breast cancer and ovarian cancer specifically. And so we thought to tackle looking at the placenta in terms of how can we better understand the placenta in this area, particularly pertaining to placental cholangiomas and its benefits, potentially looking at other cancers related to placental tissue. And now this is a quite large diagram of the many pathways associated already with the placental tissues and cancers of the placenta. Too much to go into here for this briefing, but I'd just like to highlight a few things of key concepts like angiogenesis, as well as cell cycle and apoptosis and oxidative stress that are already strongly related with other things, and the overlap this has with these placental conditions like preeclampsia and intrauterine growth restriction. The highlight taking away that we believe by understanding some of those other conditions, we can also get a good understanding to start tackling what's happening with cholangiomas specifically. And again, another large network to go into, more so to highlight at the disease level that the relationship of cholangiomas to things like preeclampsia, as well as many other forms of cancer and some other fetal conditions, right? Like hydrops, and not just that, but conditions like congestive heart failure for the mother as well as the fetus. So there is a lot that we believe we could gain by looking at this heterogeneity and getting a better understanding of that, particularly around cholangiomas. That would have greater benefit. So the heterogeneity is difficult because the placenta itself actually presents more like a cancer, right? It looks for vascularization and rapid growth. It serves a very important purpose very rapidly, right? It develops quite quickly, and then but it's only used short term. So understanding this heterogeneity would be important in differentiating between healthy placental tissues versus a cancer like cholangioma. Unbeknownst to us at the start of this project, that there's actually quite a lack of public data, as well as not enough studies done in this area. So it's already an underserved population, as well as then there's a lack of study in it as well. And even more surprising is the fact that from our literature search, we only found 15 publications since the mid-'80s, looking specifically at applications of AI or machine learning techniques to better understand this heterogeneity of the placenta, particularly around cancers like cholangioma, or just cancer in the placenta in general. So this is where we got the idea of how can we better understand some of the heterogeneity of the data sets we could find, and use that and help that apply and better understand the placenta, and then also cholangiomas in this context, using several approaches from AI and machine learning techniques. So one of the first large data sets we found was around an RNA-seq microarray differential gene expression analysis. And I don't have too much time to go into the experimental design here. It started with your general RNA-seq data processing and looking at differential expression, but then with the results of that, taking those to some of your more classical tools, looking for co-expression and gene network analysis, but then also seeing how we could use more novel and new AI techniques like pager, human-based, to connections, maybe something latent that wasn't known before, not just looking for the baseline of the heterogeneity of the placenta, but also that would be directly relevant to cholangiomas, knowing that preeclampsia and other things like oxidative stress and hypoxia are directly related to this type of cancer in particular, right? And then again, coming back and checking out what's in the literature there, even though we already knew this is an underserved area, an understudied area. On top of that, we also ended up working from another gene expression analysis, looking particularly between healthy controls and samples with preeclampsia from microarray data. And we went and trained a machine learning model off of that, looking at features like blood pressure, as well as proteinuria, and combining that with all the gene expression data across all genes looked at on the specific microarray. And here we're showing a Shapley plot of the top features relevant to differentiating between healthy control versus those with preeclampsia. And it resulted in some interesting findings, gene features that came very high to the top related to apoptosis pathways, again, looking at cell cycle, and also structural things around collagen and many other interesting things related to these pathways. So this gave us a good understanding that this was producing something. And a nice control was looking at other features, not gene specific, that blood pressure, diastolic and systolic blood pressure, was associating highly with determining preeclampsia versus healthy control. Kind of a nice confirmation that the model is picking up and detecting things and giving a little bit more weight to the results of that. Following that, we ended up looking at the heterogeneity of the RNA-seq results as well, not just saying, hey, over these time points of the sample collection from that RNA-seq analysis that we first talked about, but the heterogeneity of how things changed over several time points of collection across a 24-hour period, from like a 1 to 4 to 8 to 24-hour time sampling, highlighting the rapid changes in expression from that RNA-seq analysis. The output from that RNA-seq analysis, we then took to human base, which is applying a novel machine learning methods to looking at tissue-specific functional module detection, right? And so this is looking at, hey, across these time points from the RNA-seq analysis, what is showing up quite interestingly. And of particular interest is it brought out some very interesting networks looking at the proto-oncogene June, as well as we worked from that first time point to some of the later time points, some really other interesting proto-oncogenes and oncogenes came up. Ultimately, we ended up finding 4 relevant genes across that 24-hour time period, all directly, 3 of them proto-oncogenes, 1 an oncogene, particularly, again, focusing around some of those pathways that I had mentioned previously in the other slides. And I just wanted to highlight here more so between all the oncogenes that we looked at here, there's quite a difference in expression across these time points.
You have about a minute left.
Sure, thank you so much. Almost done. Just briefly highlighting again, coming back to that, then we went back also and took that data back to larger network analysis and other genes like FLT1, showing up from both machine learning and a greater network analysis across those 2 data sets, and other things like RBPMS, again, coming back to apoptosis, as well as other cell cycle and signaling pathways, just reinforcing the results from those findings. So what we believe we have here is a strong case for finding some of those things that are directly related not just to cancer, but also to the heterogeneity of the placenta in general, and using things like these preeclampsia data sets we know are already associated with that, and already kind of establishing, hey, preeclampsia, there is this really strong association, and maybe these are some of the indicators of that with respect to cancer. Particularly our focus looking at cholangiomas, but also definitely applying to those other ones as we saw above. And the real big thing here is to highlight that we could use this to start tackling some of these disparities that we showed at the beginning, and the impetus for this project, using these AI and machine learning techniques and our findings, looking at things for pre-diagnosis, right? Biomarkers, things to be detected during pregnancy, other things around, hey, how can we tackle this or alter treatment during the course of time, increased monitoring, also even some stuff around drug targeting, for instance, like that, in hopes to tackle this disparity and absolve this highly underserved population of this issue. And with that,
all right, thanks. So unfortunately, we're out of time. We don't have time for questions, but thank you for the presentation. And last but not least, we have 2M precise. I think that's kind of a pun there. Let's have I'll mute and let you finish out the showcase.
Can you see my screen?
Yes, it's just do the slideshow. I think you'll be good.
Okay, perfect.
Looks good.
So hello, everyone. We are a team of 5 people, and we called M-Precise. It was inspired by the concept of precision medicine, and it corresponds to the goal of our project, which is using artificial intelligence instruments and coding to develop the system which would help predict clinicians the clinical outcomes in cancer patients and adjust the treatment course based on cancer patients' clinical parameters and also gene expression. And we have chosen leukemia as a cancer model for our project. So why leukemia? particularly acute myeloid leukemia, it is very devastating disease with overall 5-year survival less than 30%, and it is even lower than 10% in elderly patients. And this is actually the disease of elderly patients. And our goal was to connect clinical parameters such as patient's age, gender, mutations that are found in malignant cells, and also treatment regimen and gene expression in immune cells, which is an integral part of leukemic microenvironment. Connect them with clinical outcomes, which are either remission or relapse. Ideally, we would like to use this connection to build the model that would somehow predict the most beneficial treatment and provide kind of recommendational system for oncologists or medical professionals identify the beneficial treatment for particular patient with particular parameters. So why, and we focused on neutrophils in particular. Why on neutrophils? Because they are basically the most abundant immune cell population in humans. They are found in all cancer patients because people just cannot live without neutrophils. So it would be technically easy to obtain gene expression data from this cell subset. And also it's becoming more and more clear that immune cells play very significant role in driving cancer and including leukemia. And here is our data set. This is a published data set available on GEO. And it includes single cell RNA sequencing data on 40 patients newly diagnosed with acute myeloid leukemia. And as I said, we wanted to identify gene markers in neutrophils that associated with poor prognosis or favorable prognosis in patients with all the diverse clinical parameters and treatment courses that they had. So I will talk a little bit about the data itself. So we used following clinical parameters as an input for our predictional system. One of the most important are age group, gender, mutations in malignant cells, therapeutic regimen chosen, treatment group, and of course the clinical outcome. The gene expression data were pre-processed by the authors of this paper, and it contained all the list of differentially expressed genes in neutrophils for all the patients. So we used that as an input to identify these gene markers in neutrophils. A little bit more about clinical data. We revealed mutations that were predominant in our patients. Here's the list. We also stratified patients into 2 main treatment regimens that include first-line chemotherapeutics, plus or with or without anti-metabolite drugs. And also we revealed that we had quite high age diversity in our cohort, and elderly patients were predominant. And with that, I would like to pass it to Rahul, who did a substantial coding part. Rahul.
Hello, everyone. As you have been briefed by Valeria, I'm going to describe the experiments that I did. Based upon the problem definition, we are actually looking at the classification problem where we are trying to identify, in the first experiment, we are trying to identify the neutrophil outcomes, which
I'm not sure if it's just me, Raul, but it's very quiet to hear you. You seem quite quiet.
Can you hear me now?
We can, but it's still quite quiet, but it's probably fine. Just didn't want people to miss.
Yeah, just talk loudly. I think the speaker might be muted because right now I don't hear anything.
Is it audible now?
Oh, yeah, I can hear you now.
Okay. So I'll continue. It's a classical case of classification problem. As Valeria mentioned, we have two classes in the neutrophil outcomes, relapse and remission. So based upon that, we perform the experiments. As you can see on the right, we have quite high results. I'm more interested in recall because if a patient that has the chances of relapse and we say that remission will happen, then we are in problem. So recall is the most important metric over here. And in the left hand side in the down, we can see a confusion matrix. The value of 691 is quite high, and we don't want that to happen. We are showing this result because we did some experiments. The results are not quite good, but it's quite significant on the problem that we are dealing with. Next slide, please. Next slide. So there are some observations that we did. If you can see on the right hand side, only one class is being classified, other class is not. So it's a problem, and that class 0 is nothing but relapse. So it gives us some insight about the problem, the data itself, that data is quite biased. Besides this, we can solve this problem by balancing the data or pre-process the data for individual mutations. This is one probability of how to solve this problem. This was the first experiment. Second experiment, next slide, please. Second experiment was quite interesting because we were trying to predict the treatment group. So this is actually part, the output of this experiment can actually be used as an input to experiment 1. It's like, it's a recommender system for the oncologist based upon the gene profile and clinical data, that which treatment group is to be applied to the patient. And we can see that here the results are quite better. Yes, so overfitting is again happening. So we have some issues with the data because we have only data for 40 patients, and the gene profiling data is actually duplicating a lot of data. So a lot of duplicate data is creating high overfitting in the data. So you can see in the left-hand corner, the confusion matrix. The diagonal with 228 and 258 gives us the actual results, but we are not interested in for a large number of values to be 327 on the upper right-hand corner. So we need to do more. Next slide, please. The same observation based upon the classification report. If you see the recall of both the classes is 0.43, 0.41, which is quite low. We need to improve the recall of this experiment as it's a case of cancer. We cannot ignore even one relapse case. So it's important for us to concentrate on this matrix. Again, why this is happening? It's a question. So we can improve the results by revisiting the feature engineering, apply deep neural networks because I think it's a complex data. Next slide, please. So to dig out what the problem is, third experiment was conceived performing clustering analysis. As we can see on the right hand side, when we plot the distribution graph, the first graph in the middle, there is a clear imbalance in the data. So first experiment we determined that there can be an imbalance in data, but on the graph on the right, we clearly see that there is a mix of the classes of remission being predicted as the classes of relapse being predicted as remission. So this is a clear case of complex data. How do we deal with this? It's again, we can use several classification different algorithms. We can apply deep neural networks, but this is a significant problem. And if we take the same approach, we can build a very good recommender system that can be helpful to recommend therapeutic regimes or even classify the patients based upon the outcomes. Over to you, Valeria.
I believe this is a novel approach for our purpose that we created our algorithms to find the most significant difference between the remission patients and the relapse patients upon analyzing the genes associated with the outcomes. The challenge was occurring in the genes of patients when all of them came in as different lists of genes. Some might show up only once, while some can be multiple times. After bringing down the complexity, we found that the same genes which were existing in both the patients of remission and relapse. And therefore, we have an opportunity to pull out the difference value of average log FC. We have plotted the graphs for 4 kinds of genes. The output shows that the neutrophil zone may carry the most essential message which decides the patient's goals, remission, or relapse. The results have strengthened the previous conclusion that the neutrophils is our focus. Next slide.
Yes, thank you, Liba. So as Liba has mentioned, he identified the genes that are differentially expressed between relapsed patients and patients who underwent remission. So we found that neutrophils neutrophils actually matter for defining if patient will go to the remission or relapse. And some of the genes that were most differentially expressed were shown here. And for example, this heterodimer is a known inflammatory biomarker. It's associated with other inflammatory conditions, and it is known that cancer is actually sort of a chronic inflammatory condition, especially leukemia. And I also used the gene list identified by Libo to run some pathway enrichment analysis, used the Enricher online tool. And I found that the pathway that was enriched in relapsed patients was neutrophil extracellular trap formation. And this is very inflammatory process that is induced by infection or cancer, and it actually creates much more inflammation and damages surrounding tissues. So based on Liba's part, we concluded that probably we should filter and narrow down our gene list to the genes associated with inflammation, and use this gene list in the classifier that Rahul built, and probably it will solve some data complexity problem. So some of our conclusions: we identified candidate marker genes in neutrophils associated with high-risk AML, and we built 2 classifiers, as Rahul explained. And as I would think the biggest shortcoming here is the complexity of the data, and probably too low number of patients to produce some significant results or some trustable results with such classifier. And with that, I would like to thank you everyone, all all the organizers and our team, Liba, myself, Rahul, and our mentors, Dr. Priyanka Gupta and Dr. Ransom Egg. Thank you.
All right, thank you very much, everybody. Sorry for running over, and I thank everybody for sticking around to the bitter end. Looks like most people did. And now everybody worked hard, and now take a break. Our judges will now have the hard work ahead of them of evaluating all these presentations. If you missed some of it, and you can, the recording will be posted soon. All right, and I'll be in touch with the judges. And be sure to attend our closing ceremony and awards presentation next Friday at 10 o'clock. All right, thank you, everybody. Have a great weekend, and thanks again for all your participation and hard work. All right, bye-bye now. I'm gonna turn off the recording and stop the meeting.
Thanks, Amy.